Have you ever wished you could just rewrite a typo in the book of life? What if we could correct genetic diseases, create supercrops to feed the world, or even wipe out pests like mosquitoes? It sounds like science fiction, but it's happening right now, thanks to a revolutionary technology called CRISPR. And in the next 10 minutes, we're going to break down exactly what it is, how it works, and why it's poised to change everything. So, what is this thing with the funny name? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. I know, it's a mouthful, but you don't need to remember that. Think of it like this. CRISPR is a natural defense system found in bacteria. For billions of years, bacteria have been fighting off viruses. When a virus attacks, the bacterium captures a small piece of the virus' DNA and stores it in its own genetic code. In these clustered repeats, it's like a genetic most wanted gallery. If that same virus attacks again, the bacterium uses a copy of that stored viral DNA to guide a protein to the invader and chop it up. It's a precise molecular search and destroy mission. Scientists realized they could hijack the system. They figured out how to create their own guide, a small piece of RNA to match any DNA sequence they wanted to target. This guide RNA is then attached to a molecular scissor, most famously an enzyme called Cas9. So, the process is surprisingly simple in concept. You have your guide RNA that knows the exact address, and you have your Cas9 enzyme ready to make the cut. You put them together and introduce them into a cell. The guide RNA zips through the cell's DNA until it finds the perfectly matching sequence, the spot you want to edit. Once it locks on, the Cas9 enzyme cuts the DNA, creating a clean break in the double helix. Now, this is where the magic really happens. A cut in the DNA is a cellular emergency. The cell immediately rushes to repair the break, and we can take advantage of that natural repair process. The simplest thing we can do is just let the cell fix it on its own. Often, the cell's repair is a bit sloppy and makes a small error, which effectively knocks out or deactivates the gene. This is incredibly useful for studying what a specific gene does. If you turn it off and see what happens, you learn its function. But what if you don't want to just break a gene? What if you want to fix it, or even add something new? Researchers can provide a new piece of DNA as a template. When the cell goes to repair the cut made by Cas9, it can use this new template to patch the gap. This allows us to correct a faulty gene that causes a disease, or even insert a whole new gene with a desirable trait. Imagine replacing the single misspelled letter in the gene that causes sickle cell anemia with the correct one. That's not a hypothetical. It's a therapy that has already shown incredible success in clinical trials, essentially curing patients of a lifelong painful disease. And the toolbox is getting even more advanced. While Cas9 is the original star, it's not the only player anymore. Scientists have discovered other enzymes like Cas12 and Cas13. Cas13 is particularly cool because it targets RNA, the temporary messenger molecule, instead of the permanent DNA blueprint. This allows for temporary edits that don't permanently alter the genome. Then you have technologies like base editors and prime editors. Think of these as the next generation. Instead of making a full double-strand cut, which can be a bit risky, they work more like a pencil and an eraser. Base editors can chemically convert one DNA letter to another without breaking the DNA backbone. Prime editors are even more versatile, allowing for small insertions, deletions, and all 12 possible letter-to-letter swaps, with incredible precision and fewer risks. They are rewriting the code of life with unprecedented control. Of course, having these amazing molecular tools is one thing. Getting them to where they need to go is another. This is one of the biggest challenges in the field, known as the delivery problem. How do you get the CRISPR machinery into the trillions of cells in a human body, or even just the specific organ you need to treat? Scientists use a few clever methods. One way is to use a disabled virus, called a viral vector. They strip out the harmful viral genes and replace them with the CRISPR components. The virus then does what it does best. It infects the target cells and delivers its genetic payload. Another popular method is using lipid nanoparticles. These are tiny bubbles of fat that can encapsulate the CRISPR system and merge with the cell membrane to release their contents inside. It's the same technology used in the mRNA COVID-19 vaccines. For cells that can be taken out of the body, like blood stem cells, they can use a technique called electroporation, which uses an electrical pulse to temporarily open pores in the cell membrane, allowing the CRISPR machinery to get in. Choosing the right delivery method is crucial for the safety and effectiveness of any CRISPR-based therapy. This brings us to the critical topic of safety and accuracy. What if the CRISPR system makes a mistake? What if it cuts the DNA at the wrong location? These are called off-target effects, and they are a major concern. An unintended cut could disrupt a healthy gene or, in the worst-case scenario, activate a gene that causes cancer. Researchers have spent years developing more precise guide RNAs and new generation editors to minimize these risks. Another challenge is mosaicism. This happens when the editing process doesn't work in every single target cell, leaving you with a mix or a mosaic of edited and unedited cells. For some diseases, editing just a portion of the cells might be enough, but for others, you need a much higher success rate. Rigorous testing is essential to ensure these therapies are both effective and safe. Despite these challenges, the medical applications are moving forward at a breathtaking pace. We've already mentioned sickle cell disease and a related blood disorder, beta thalassemia, where editing a patient's own blood stem cells and returning them to the body has produced remarkable results. Clinical trials are also underway for a type of hereditary blindness, where CRISPR is injected directly into the eye to correct the faulty gene in retinal cells. Scientists are also exploring how to use CRISPR to fight cancer by engineering a patient's own immune cells, called T cells, to make them better at finding and destroying cancer cells. Others are investigating its potential to treat high cholesterol by editing liver cells, or even to eliminate persistent viral infections like HIV by cutting the virus's DNA out of our own cells. The impact of CRISPR extends far beyond human medicine. In agriculture, it's a game changer. Scientists are using it to create crops that are more resistant to diseases, drought, and pests. This could dramatically increase food security and reduce the need for chemical pesticides. They're editing mushrooms that don't brown, tomatoes with improved flavor and shelf life, and wheat with higher yields. And it's not just plants. In biotechnology, microbes are being engineered to produce biofuels, sustainable materials, and even new medicines. There's also a more controversial application called the gene drive. This could potentially be used to alter an entire species in the wild, for example, by making mosquitoes unable to transmit malaria. While the potential benefit is enormous, the ecological risks of releasing such a powerful technology into the wild are still being heavily debated. And that leads us to the big ethical questions. The conversation around CRISPR is often divided into two categories: somatic editing and germline editing. Somatic editing targets the regular cells of the body, like blood, liver, or eye cells. These changes affect only the individual being treated and are not passed on to their children. This is where all current clinical trials are focused, and it's generally viewed in the same ethical light as any other advanced medical treatment. Germline editing, on the other hand, involves editing sperm, eggs, or embryos. These changes would be heritable, meaning they would be passed down through all future generations. This is a profound and permanent alteration to the human gene pool. For this reason, germline editing is currently banned or strictly regulated in most countries. The debate is complex, touching on questions of safety, consent, and what it means to be human. Finally, let's talk about cost and access. Right now, the first CRISPR-based therapies are incredibly expensive, costing millions of dollars per patient. This raises huge questions about equity. Will these be treatments only for the wealthy? The hope is that as the technology matures and competition increases, the costs will come down, just as they have for other revolutionary technologies like DNA sequencing. In the research lab, CRISPR tools are already remarkably cheap and easy to use, which is why the field is exploding with innovation. Scientists around the world are working tirelessly to make these therapies safer, more efficient, and ultimately accessible to everyone who needs them. From a bacterial defense mechanism to a tool that can cure genetic disease and reshape agriculture, CRISPR is one of the most significant scientific breakthroughs of our lifetime. It's giving us the ability to not just read the code of life, but to rewrite it. The journey is just beginning, and while we must proceed with caution and thoughtfulness, the future it promises is nothing short of extraordinary. Thanks so much for watching. If you found this video fascinating and want to learn more about the cutting edge of science and technology, be sure to hit that subscribe button and tap the like button so you don't miss our next video. We'll see you in the next one.